microbiological and nutritional qualities of locust-based bacteria in the bolster using Lactobacillus plantarum CIP103150 and the Lactobacillus fermentum CIP1080 as starter culture. The aim, go to the aim. The aim of the study, check the microbiological and nutritional qualities of Pachyderm bilobosa produced using identified strains of lactic acid bacteria, Lactobacillus plantarum CIP103150 and the Lactobacillus fermentum 102987 as starter culture. The objectives were: 1. To produce improved locust-based using known strains of lactic acid bacteria, Lactobacillus plantarum 103150 and the Lactobacillus fermentum 102987 as starter culture. The second objective was to assess the microbiological quality of the improved locust base produced. The third objective was to determine the plasmid composition of the locust base produced. The fourth objective was to determine the mineral composition, pH, and ascorbic content of the improved locust base produced. The fifth objective was to check the amino acid profile of the improved locust base produced. The sixth objective was to determine anti-nutritional compound in the improved locust base produced. The last objective was to evaluate necessary evaluation of the improved locust base produced. Please go to optimization production. All of these objectives were achieved following standard microbiological and NOC standards. Optimization production. Iru is a product, locust base is a product that has been here even before my own parents were born. We consume it, but however, we have not explored the economic potential because we have not improved it. It's still on the local level. So what I did was to actually improve the production process using aseptic microbiological techniques. The first thing, 100 grams of Iru locust base. 100 grams of locust base was boiled at 121 degrees Celsius for 2 hours to obtain the net shell because it's coated with 2 shells. Another one at another 121 degrees Celsius for 2 hours to obtain the cotyledon. The cotyledon, which I washed aseptically and allowed to cool to 40 degrees Celsius before I inoculate my starter culture at ratio 1:1:5. Please go to the results. This was the first result, which is of course the first objective of the study. Different media were used in the microbiological assessment, starting from tryptic soy agar, which is a rich multi-purpose media. From this result, you can see that the M sample, which is the improved, thank you, sir. The improved cycle, which is a sample M, recorded 2 few to count for tryptic soy agar. While the sample C, which is the commercial spontaneous product, that is a traditional method of protein fermentation, yielded too numerous to count for a CFU for the product. And likewise, in the case of selective media like SDA, Saboreal distress agar, to detect fungi, this result shows that the improved sample M, there were no detectable fungal growth in the sample improved, while sample C, which is the commercial sample, recorded 4.0 times 10⁻³ as compared to the improved sample. However, in EMB, no detectable microbial growth were observed both from the improved sample and the commercial sample. This result is in conformity 2018, which says that lactic acid bacteria has competitive exclusion potential, which make them to produce a true acidification and able to produce acid microbial metabolites such as bacteriosis and hydrogen peroxide, which make the medium unfavorable for unwanted or targeted microorganisms by lowering the pH of the medium to eradicate the spoilage organism. This result shows that. Proximate composition. This proximate composition from here, we can see that sample U recorded high percentage of moisture content, which of course do not support longevity, that is shelf life of the product. And while sample C was nearest to this sample U, however, the improved sample, which is sample N, which is the improved sample using these two strains of organisms, recorded least moisture content from this. And also coming to the crude fiber, crude protein, the highest percentage of crude protein was recorded for the improved sample, which is an N, and as well as crude fiber and other proximate composition. This result also is in conformity with Olukomaya's research in 2020, which says that low moisture content supports longevity, that is long shelf life of the product, and high protein enhances assimilation of protein digestibility and assimilation. And overall, according to the work of Atari 2019, this work shows that indeed fermentation, especially controlled fermentation, increases the nutritional quality of fermented products. pH. Okay, this is the pH of my research. From this research, you can see that sample 5 recorded highest number of pH, while sample M and sample C 6.2 each were equal. This result is also to show that low pH indicates high level of preservation, that is longevity in shelf life, which ought to be attributed to fermentation, while high pH could be attributed to negative microbial interaction that in cause of fermentation. Mineral composition. Thank you. This is the mineral composition. From this result, we can see that both calcium, zinc, iron, potassium, they were all improved in sample N. They were all enhanced with the highest percentage of mineral composition, while sample C recorded the least value of mineral composition in most of the parameters. This result is also in conformity with Agebo Test 2021 that says increased minerals is attributed to enzymatic breakdown of anti-nutritional compounds found in food, while reduction in mineral composition can be attributed to nutrient leaching during fermentation. And this also confirms with why I have low moisture content for my proximate, because when water is lost, there is possibility of reduction in some of the nutrients. So please go to it. Amino acid profile. Indeed, this work really shows the uniqueness of fermentation in food products, as all the essential amino acids were all enhanced, improved significantly in my optimized product, M sample. And also, according to the work of Zebedee 2022, high amino acid profile can be attributed to the role of fermentation, which is naturally known to increase nutrient content of food products, anti-nutritional compounds. In order, because we are aiming at a product that we want that is consumable, So I wanted to know the nutritional compounds that will be present in this product. Fortunately for me, I realized that there was no significant records of antimicrobial nutrients. This nutrient that can interfere with the body and system to neutralize the essential nutrients present in food. So, but however, we can see this which is about 160.50, which was recorded for us for the commercial product from the uncontrolled or spontaneous fermentation. So this result also confirmed with the work of Kashiri 2012 that the difference recorded, or maybe the result, the significant increase in the decrease in the nutritional compound can be attributed to enzymatic absorbency in the course of fermentation. I can therefore say that this work indeed controlled fermentation necessitated the detoxification to enhance microbial safety and nutritional value. Ancestry evaluation. This product was widely accepted, the improved product was widely accepted by all the panels of judges, 60 panels of judges using hedonic scale, except the aroma, which I understand, because they are not used to this improved product. They are still used to the traditional smell of food. But aside that, if you check every other parameter, texture, color, the overall acceptability, they all accepted it except the aroma. Maybe it's still new to them, which I understand. Which of course, according to the work of Adebute 2017, that fermentation can produce more alkylic products. Recommendation. This work has achieved an improved locus base, which using lactic platyvibacillus, no stress of lactic acid bacteria, lactic platyvibacillus plantarum, CIP103150, and the Mosin lactobacillus fermentum 102980, which I isolated during my master's degree, and it's now been documented with NCBI in Maryland, USA. So this product has achieved, aside that we have achieved the non-starter culture, I also achieved microbiological improvement, nutritional, amino acid profile, mineral composition, and sensory evaluation, improved sensory evaluation of this product. Recommendation. My humble recommendation at the end of my research. Number one, large-scale production of Iru using lactic platyvibacillus plantarum CIP103510 and the Mosin lactobacillus fermentum CIP1020 should be encouraged to boost the economic potential of the product. Number two, owing to the potential of the two probiotic strains, commercial starter culture formulation should be developed and publicized to create awareness and more local processing in the ultimate goal of standardizing method of production. Recommendation 3. Local producers should be trained on hygiene and aseptic techniques that will ensure the microbiological safety and wide acceptability of Africa-originated fermented food. Thank you for listening to me.
Then if you go to your page note, page 5B, where you did acknowledgement, you took me to a puzzle after acknowledging your supervisor, Professor Martin and myself. And the next person you acknowledge in the department is Dr. Stephanus, Haruna and McKenna. I was so shocked that you don't know that we have a hierarchy. After your supervisors, we have 5 other professors in the department. We also have head of department. You never recognize any in your acknowledgement and you are appearing before us. It's a very serious error. You go and put it twice and remember the hierarchy and put it in order. I also saw something that marvelous me under your acknowledgement after acknowledging Dr. Stephanus, Haruna, and Penny. you say Professor Adamu Akiala, Ishaku Akiala. It baffles my mind. Because there are a lot of controversy.
Then we can step down from the proposal.
Please. Find out the correct because we are appearing before academicians and we are not politicians. We do what is definite fact. Akiala has not been pronounced a professor in National State University. So it's a erroneous for you to come and present him as a professor. If he's a professor elsewhere, it's not yet confirmed in our department. If the head of department is here, the dean of faculty is here, so it's an error. So you have to expunge it in your work. Note it and expunge it, right? And then recognize other professors in the department. They are the people assessing you. Okay, you had a comment that you should be stepped down. Don't take it for granted. After doing a beautiful work, now technicalities will not be enough. Please, yeah, it's not good. I've said before that you will make a good attempt, and I think you are getting the degree. But this small thing, don't encourage it, please. Thank you so much. Sorry, the panel of judges refers to who were the panel of judges that accepted the vote? As presented, the 60 panel of judges. So when you see people that did sensory assessment, no, sensory assessment is done with panel of judges. You select people.
Then I explain. I'm interested.
The panel of judges, there's a form that is a hedonic scale, that is on the scale of 1 to 10. So you gather these people just like you are sitting in this room, randomly. You show them the product. You will not tell them which one, is which. So they check the texture, the color, the taste, and all of that. They use it to rate it. This is how I got that.
The report.
Sorry.
Okay.
Thank you very much. I think the first time I was saying you that you don't use yellow coloration for PowerPoint. It doesn't use the contract at all. I don't know where that color yellow came from, right? And it's of course using it. It doesn't should be contract. So avoid that. Your sample, the local bean you produce using this your organism. You did the fermentation in the lab. Was it controlled fermentation? Was it laboratory controlled fermentation?
What I did is that I
Just
aseptic procedure.
in the lab.
Yes.
It was laboratory control. Why didn't you also do the laboratory control with the one without inoculation? Why did you have to go and then get from the locals? Why didn't you subject both of them under the same condition? Because you are comparing the two. So if you did laboratory control with one, you should also do the laboratory control with the one that was not inoculated. You replace them, don't allow bias. Because what the people are doing outside there, it is not controlled. I know that when you are targeting it, because people feed on that. I know that was your. But if you really want to control in terms of their involvement, then both of them should have been laboratory controlled. The one inoculated with your this and then the other one, the normal. Both would have been laboratory controlled. And then, secondly, how did you confirm that even the one that people use, the one you bought from that Kapusi, did not have this organism? Because these organisms are involved in fermentation. You know that? So there's a possibility that they could also be found in the sample that you went and collected Kapusi. Did you try to find out whether they were there or not?
Yes, during my previous research, during my master's, before I even took interest in this, I realized that there are strains of lactic acid bacteria actually in the commercial product. like the pediococcus, the stock, and the rest of them. So what I was more concerned is using a known strain of lactic acid bacteria in the fermentation process.
It's okay. It's okay. It's okay.
was done in stages. She isolated those organisms that she used at the onset during her MS.C. and was able to identify them properly from the local production process. And the gene bank was able to distinguish these isolates as variants of the type strain. And in this stage, even though you make a very valued contribution that there should have been a control, but it has gone to the point where the comparison is between the work done in the lab and the local.
Yeah.
And that is the result she is presenting. Because I think there was an ambition towards the production of the she should not because she's learning, not it when learning it. There should be another control.
Subject them to the same conditions.
The high point of this work is that she has already done it, and even beyond the objectives, achieving the objectives, because she has the product. I would have, but I want to challenge her so that we get to the next level, where we will now get that approval and brand it and commercialize it. But the question now is that what is the volume, what will be the volume of production per time, per week? It's not part of the work, but we have to look at that so that we determine the scalability and the commercial potentials. So these are just, but not it as a student that there should have been a control. But even if there's no control, the results is elaborate enough to speak for itself. Thank you very much. But not this as a student. Thank you.
Round of applause for her. The next is Paul Adam.